And thanks for it. Thanks to Natalie. I actually went to grad school with Natalie, so we go back a little ways. But it turns out that a couple of the people here that are Michiganders, and I didn't know that. I'm from Michigan myself.
Wow. Okay.
Small world. Okay, so welcome everyone, and I'm going to turn it over to Elaine.
Okay. Thank you, everyone, for coming. This is a very well-attended meeting. I'm going to give you some background on Natalie, but before I do that, I want to thank the volunteers that made this meeting possible. First, I can see Ann Taylor, and she's going to write this meeting up for the Vortex. Okay. And then I can also see who do I see? Fanny. Fanny is recording it, and she uploads the recording to our YouTube channel. Okay, so without them, that meeting will not be as well chronicled. Then the other thing is the flyer that you got was put together by another volunteer, Sheila Ye, and then another volunteer, some of you will have contact with, named Tony Meow from Chevron. The volunteer that's on the screen somewhere, Michael Chang, where are you? Raise your hand. Michael is, he's the director of our contest, okay? And the contest is absolutely a volunteer activity. It is based on three multiple choice questions from the speaker's talk. So if you'd like to participate and get a chance to win prizes, you can do that at the end of the meeting. Then if you win, Tony Meow will send out the prizes to your house, but I have to collect your home address. And I won't give it to anybody else. All right. Now I want to talk a little bit about Natalie. Natalie is a chemist trained at Stanford University for her graduate work, and she studied organic chemistry. In graduate school, you get to concentrate on a particular area of chemistry. So, in organic chemistry, what chemists do is they make molecules and then they verify their structure by using all kinds of analytical techniques. So, that is in part what Natalie did as a graduate student. Then, at the close of her graduate education, she went to work for Syntex Research, where she studied process chemistry regarding prostaglandins. Now, that's really stuck out to me because I put prostaglandins in my eyes every day. And I know that prostaglandins are not the most stable compounds in the world. And so now her job was to make tons of this material. That's what process chemists do. Tons of a material that is kind of unstable. That's a hard assignment. Okay. Um, then I wanted to also point out that Natalie was a pioneer. And I don't mean the kind that came over in a covered wagon. I mean the kind that really makes a difference in women chemist circles. Okay. When Natalie was a graduate student at Stanford University, there were not that many women graduate students. Okay. The other thing is when Natalie was studying organic chemistry as a graduate student, there weren't that many women studying organic chemistry. And in fact, even today, organic chemistry is not exactly the most popular area for women graduate students to be studying. That is why there are some groups that concentrate on organic chemistry and the participation of women. The third thing where she was a pioneer is just simply being hired at Syntax. Okay. At that time, it was like the beginning of the era when women were actually being hired at various places, Syntax, Chevron, places like that. Okay. So she was a pioneer in her own right. And then while she was at Syntax, she decided to pivot toward this drug regulatory affairs area. And she participated in that for many, many years. And she served on panels. She did teaching. She does consulting now. And so she's done a lot in that area, the area she pivoted to. Then finally, uh, Natalie is a volunteer extraordinaire in the ACS. She is one half of the team that is putting together the Western Regional Meeting. Okay, so I'd now like to turn it over to Natalie. And if I missed any of your really important contributions, let me know and let the audience know. You can brag a little bit because you're such a pioneer. Okay?
Okay, well, thank you. I'm going to share my slides, and so you can see them all. Thank you, Elaine, for that lovely introduction. I have a lot of slides, so I won't go too far, but just for some of the women and younger women in the audience, when I was interviewed for a job at Syntex, I was actually asked, does your husband approve of this job? And what are you going to do when you get pregnant? Which was assumed to happen. I turned around and reported that to the HR department because those are two questions that even at the time were completely illegal. And they hired me anyway. Maybe they had to hire me after all that because they didn't want to face the consequences of that. But I had a good career and I enjoyed it. And the environment, other than those questions, in general, the environment was quite comfortable. Okay, can you all see my slides? I started, as several people have mentioned, we're doing the Western Regional Meeting, and so a little bit of shameless promotion for the meeting. It's coming up. It'll be in October in San Jose, California. It's a joint meeting between the California section and the Silicon Valley section. We are anxious to see you all there. There will be a focus on undergraduates and an opportunity to present their research and some workshops around how to survive graduate school and how to keep your career going. There will be a lot of scientific presentations, and so I just hope to see you there. The abstracts are open. The call for abstracts is open. The call for awards, if you want to nominate someone for awards, is open. Go to the website. You can just scan that URL or that code, QR code.
Let me say I put the website in the chat as well.
Thank you. Okay, so I'm going to talk about drug development from the laboratory to the marketplace. I have a bunch of slides, so I'm going to rush through them so that we aren't here all morning. But if you have questions, just speak up. I'm not good at always checking the chat or seeing the hands being raised. So if you've got a question, just say something about it. And I thought I'd start. I'm going to try and keep politics out of this, although it's a little hard these days. I thought I'd start with how are drugs regulated? Because as Elaine mentioned, my career after the initial time as a chemist switched to being in regulatory affairs. So drugs are regulated through the Department of Health and Human Services, which at the moment is the FDA is current secretary is I'm sorry, the Department of Health and Human Services current secretary is Robert Kennedy Jr. The FDA is a guy out of head of FDA is a guy out of Johns Hopkins named Makary. It is a huge department, $1.6 trillion budget, 83,000 full-time employees, at least before the current administration took over. I don't have current numbers. And this is who's in the Department of Health and Human Services. And the reason I bring this up is that I wanted to point out that there are multiple organizations that cover regulating and developing drugs, and they're discrete and different. So there's the FDA, who approve the drugs before they can be sold and who work on the market, the support of the clinical trials and clinical research. There's the NIH, who is not the FDA. You know, they do sponsor a lot of research, sponsor a lot of clinical trials. And during the COVID period, we heard a lot about NIH. That's where Fauci was part of NIH, Dr. Fauci. FDA, excuse me, FDA was the vaccine guy that was the one you heard so much about during the COVID period was Marks, and he was in FDA. And then there's the Center for Disease Control, who do mostly epidemiology of checking where the disease occurrences are happening, capturing the data around that right now. Of course, there's a lot of work on measles and where the measles outbreak is going. During COVID, it was more about COVID. And then the last group that gets involved in drugs is the Center for Medicare and Medicaid, who handle reimbursement for people who qualify for either Medicare, so people who are over the age of 65, or Medicaid, which is a supporting program for people who are in need of financial assistance and for drugs. And they're distinct and they're different and they're not part one and the same. You know, Dr. Fauci could say one thing, Dr. Marks could say another thing, and CDC could issue different guidance. And I think that confused a lot of people because they thought, oh my God, we're getting all these different messages. Well, they're different organizations with a different focus. Just quickly at a glance, FDA is responsible for FDA. First of all, let me start with FDA stands for the Food and Drug Administration, not the Federal Drug Administration, which a couple of our presidents have misspoken more than once. And as Food and Drug Administration, they are responsible for the review of foods, medical products, drugs, tobacco, devices, medical devices. They spend about 20 cents of every dollar you spend goes towards something that has been involved in review or meeting regulations posed by the FDA. And they regulate most of the food supply, 78%, not meat, poultry, and some egg products. So these are the products. You know, we in most of this class, we're going to talk about our class. Sorry, I teach a lot, so I'll fall into that. But most of this lecture will be about drugs or biologics, but they also cover veterinary products, medical devices, tobacco, and then they don't do pre-market approval, but they establish safety and purity standards for foods, vitamins, dietary supplements, cosmetics, and radiation emitting devices. So even your microwave oven or your TV, if it were an old TV, not a new one, a cathode ray type TV, those have to meet standards for radiation emission that are established by the FDA. What they don't regulate is alcohol, drugs of abuse, food production. So USDA is responsible for the farming aspects of poultry products and eggs. How should the animals be housed? How should they be treated? That sort of thing. And USDA establishes dietary guidelines, you know, the pyramid or the triangle on what percentage of your food should be meat or dairy or wheats and whole grains. The other thing that FDA does not do is they don't dictate medical practice. Doctors can prescribe drugs. The drugs have to be approved by FDA before the doctors can prescribe them. But doctors can use the drugs to treat their patients however they think it's appropriate. So that means they can use them at slightly different dose regimens or doses. They may decide to reduce the dose or increase the dose based on the individual patient they're treating, and they can use them for other indications, other treatment indications, that they, as the doctor, the medical practitioner, thinks is appropriate for a particular patient. They do have to report, there's insurance issues around off-label use. Off-label means not according to the package insert that FDA has approved, and the insurance companies may raise a problem about paying for the drug, but the doctors can prescribe them and the patients can take them. So all that fuss about ivermectin and during COVID, it wasn't the FDA's fault. Ivermectin's an approved product, can be used. Hospitals, it wasn't necessarily a good idea, but it wasn't due to FDA. So the first question you need to answer if you're developing a new product is, what is your product? Is it a drug? Is it a device? Is it a food? Is it a cosmetic? And that may seem on the surface to be kind of easy, but in fact, as you peel back the onion, it's not quite as easy as it might seem. So a drug is something, or a biologic is something for use in diagnosis and all those other things, prevention of a disease or a condition, and an article other than food that will affect the structure of the function of the body. And the word drug, just in case anyone is concerned, includes the active pharmaceutical ingredient, the powder or the chemical that's made, as well as the formulated drug, the tablet or the injection or the patch or whatever. A device is all those things there, including medical software that's used for diagnosis, but does not achieve its intended purpose through chemical action. So a silly example is collagen, bovine collagen used for filling wrinkles, treating wrinkles, or puffing up people's lips. That is regulated as a device because it's not approved for its action, is not through a chemical purpose, it's just for a filler. Botox, botulinum toxin, is also used to treat wrinkles, but that does have an action through a mechanism of action through chemical action. It's paralyzing a nerve. And so that is regulated, in this case, as a biologic, not a drug. And so that just gives you an example of how you have to spend some time thinking about what your product is going to do. And FDA actually has a website called, is it a cosmetic, a drug, both, or is it a soap? And that website list is down there to give you some of the thought process you might want to work your way through. I didn't talk about cosmetics. In fact, they'll be on the next slide. So cosmetics are what you think of. They're for cleaning or beautifying or altering your appearance. And examples of things that are regulated as cosmetics are toothpaste, shampoo, hand soap, massage oil. But if you put a drug in them, like an anti-dandruff shampoo or an antibacterial soap that has something to kill the bacteria, or fluoride in your toothpaste, all of a sudden your cosmetic becomes a drug and has to be regulated as such. Sunscreen, for example, has drugs in it to absorb the UV rays. So those are all drugs and have to comply with GMP, good manufacturing practices, to make sure that they're prepared appropriately. Vitamins and food supplements is another area where you get into this gray area of, is it a drug or is it a food supplement? Vitamins and dietary supplements are not subject to FDA pre-approval review. By that, I mean they do not have to be, the data does not have to be reviewed before they can be marketed. They were exempted back in 1994 by this particular act listed here. And this would cover vitamins, minerals, amino acids, things that are intended to be taken by mouth. They do have to meet safety standards, and they do have to be listed. A manufacturer has to list their manufacturing site, and they have to label things appropriately. FDA and drug regulatory, a lot of it's about labeling. And by labeling, I mean both the label that's on the bottle or the package, but also the promotional material and the information that comes out on the prescribing information or the package insert. All of those are called labeling. And a lot of our work is on proper labeling of information. Vitamins and foods can't promote specific drug claims. Now you're going to say, but wait a minute, I hear all the time, as I watch a TV ad about take this drug, it will improve your memory or it will help your joint health. So what is allowed for dietary supplements are health claims. There's some examples, or nutritional content claims. It's high in calcium, or structure and function claims. Ginseng will help your natural energy. None of those talk about a specific disease or condition. They're just general. There is a required disclaimer on all dietary supplements. This language has not been evaluated by the FDA, not intended to diagnose, treat, cure, or prevent any disease. But they will then go on and say all of that on the bottom, but they'll then say a diet low in fat may reduce the risk of some cancers. Or some studies have shown that fill in the blank product will increase your natural energy or will enhance your alleviate the joint problems. Notice I didn't say pain. A lot of drugs, a lot of dietary supplements go way over this line. And here are some examples in the COVID field and in the opiate withdrawal field. I could have put up examples for things for erectile dysfunction. There are many of these, especially on the internet, where they talk about a safe and effective natural supplement to ease the physical symptoms or break the painkiller habit or relieve your symptoms of withdrawal or craving or to kill nasty pathogens. These are all disallowed, and FDA has an arm of FDA, which is their police or their compliance arm, will issue letters to the companies asking them to withdraw their product from the market. They withdraw their product from the market, then they rename it, then they put it back on the market. It's like a game of whack-a-mole for the FDA to keep things off of that. But you as a consumer should always question what they say, especially if it's going to prevent you from taking something else that might be more appropriate for helping you with your withdrawal symptoms or treating your COVID rather than colloidal silver or something. So now I'm going to get a little more scientific. We have a bunch of different types of drugs. They're novel drugs or innovator drugs. There are generic drugs, and those are both prescription products. You have to get a medical prescription to sell it. There is also a system of non-prescription drugs, over-the-counter drugs, which are covered through a monograph system. To be available to market on any of these categories, the drug must have been shown to be safe and effective, although the standards that are required both for proving safety and effectiveness, as well as the manufacturing standards of how they're made, differ depending on what category they fall into. Another sort of side thing that I want to go through quickly before we dive into this is drug names. You may have noticed that most drugs, any new drug, any novel innovator drug, will have two names, a trade name and a generic name. And anything you read will describe the trade name, Humira, the generic name, adalimumab, and then the description of the dosage form. In case of Humira, it's an injection for subcutaneous use. In the case of remdesivir, which you may think of for treating COVID, you may have heard it more as remdesivir, which is the generic name. The trade name is Veklury, so they should write it that way, or Prilosec, which is omeprazole for treatment of heartburn, as a tablet. And so that's the format: trade, generic dosage form. And that should be used per regulation on all of the advertising, all of the package inserts for a product that's still under, that's not a generic product. The generic names are regulated by USP. You apply for one, and they actually have a certain amount of intelligence embedded in the name. There are prefixes, and suffixes that help you identify the drug class. So there's a class of statins that are approved to treat lipid disorders, high cholesterol, and they will all end in the phrase of statin, like atorvastatin is an example. Monoclonal antibodies all end in mab, up here for Humira, and the 2 or 3 letters that precede it actually also have intelligence. MUMAD means it's a humanized monoclonal antibody. If it's an IX mab, it would be a chimeric monoclonal antibody. And so you get some information from the name itself. And this is very helpful for the pharmacist, the doctor, and even the patient if they know how to interpret it, because you can tell if you're given one statin and you don't like that one, you want to try a different one. Well, or you don't like statins at all. Well, don't get another one that is called a statin. And the last example I showed here is Ivir. Things that end in Ivir are all antiviral agents. There's also in the prefixes, they tell you the chirality of the product. Drugs that start with ESE, or sometimes there's 2 S's there, are the S enantiomer. Drugs that start with AR are the R enantiomer. And drugs that start with RAC are the racemic. Now, that's not a requirement. Many old drugs are racemic and don't have the RAC in their name. But if there's a version, if the racemic version has been chirally separated and developed separately as 2 different drugs, then they would make that name. Once a product goes generic, you only use the generic name. So Prilosec or omeprazole tablets, you can still buy as Prilosec, as a branded product, but you can also buy them as a over-the-counter generic product, and then it would just say omeprazole tablets on it. So for a product to be over-the-counter, it has to be shown to be safe and effective for use by the general public without a medical professional's advice. So the condition has to be something that can be easily identified: headache, allergies, toenail fungus, vaginal infection. We all know when we have one of those things. We don't need to go to a doctor to be told that we have hay fever, it's the springtime, and our nose is running and are itchy. We know that. So we can already identify that ourselves, the condition. The drug also needs to be low in side effects and have a wide safety margin. And a safety margin refers to the amount of drug that can be given before you run into adverse events. If you've got a wide safety margin, you know that the patient, if they were to take twice the number of tablets that they should be taking, which some people do, they're not likely to run into problems for side effects. If there's a low therapeutic index or a low safety margin, the FDA would not allow that to go over the counter because they want it to be more under the management of the physician. The OTC products are described by a monograph. So there's published in the FDA documents, the Code of Federal Regulations, and in the USP prescribing information documents, there's a monograph that several pages long that describes the strength that you're allowed to sell, the indication of use that can go on the label, the dose, specific language for labeling. And once something's in a monograph, you can come along, if you wanted to start a drug company and make aspirin, you can start a drug company and make and sell aspirin without showing that your aspirin meets the requirements. You have to list your manufacturing site on the FDA website, but then you could just market the product. And just to make this a little clearer, here's a picture of loratadine tablets. which is just called allergy relief by CVS. And here, this language of sneezing, runny nose, itchy, watery eyes, that's prescribed in the monograph. They can't add to that itchy ears if they wanted. That would be now misbranded or inappropriately labeled. And then on the backside, it tells you the strength that's allowed is prescribed, the temporary use. I mean, here again is the same language as on the front, and the storage conditions are prescribed, the directions for use are prescribed. All of this language is required and listed, but then now you're free to sell your loratadine tablet at CVS. Questions on any of that? Okay. Drug development is a risky proposition, and you, as chemists, probably all know this already. They've estimated that it takes between 6 and 10 years from the identification of a new drug to a product that's actually out in the marketplace sold. And of course, these estimates vary widely from development strategy to type of product, but they're, give or take a couple years on all of these, they're about right. And it's often referred to as a funnel. They say many, many, many compounds are developed in drug discovery, and from that 10,000 plus, this particular Millikan Institute review estimated that 250 went into preclinical testing, from which we got 5 drug candidates that went actually into humans, from which 1 product ends up being approved. And I'll go through what is involved in the different steps as we go along. Here is another cartoon showing what goes on during the funnel. This is all the top part of the funnel, but there's, the chemists synthesize the drug. They make enough to be tested. It gets screened in biology. There's an assay that's developed so that we can monitor it. There's an assay for the preclinical. The preclinical means animal testing, animal and test tube testing, in vitro testing. There'll be an assay developed over here by the chemists to determine how to properly determine what the strength and quantity you've got, the purity of your drug. From that, it goes into animal testing. ADME stands for absorption, distribution, metabolism, and excretion. So we have to study first in animals how much of the drug is absorbed when it's given by an oral tablet or a subcutaneous injection. How is it distributed? Does it end up in the fat? Does it stay in the bloodstream? Does it get to the brain? Does it accumulate in the liver? How is it metabolized, if at all? And how is it excreted? Those two go hand in hand. Some drugs are excreted unchanged. No metabolism occurs at all. Some drugs have to be metabolized before you have an active drug. Those would be called prodrugs. So all of this work gets done on the 250 drug development candidates that have been identified before you go into clinical development. This is a different schematic showing you a little bit more of what happens, but the organic chemist at the very beginning and the pharmacology research people identify the target they want to hit, the particular enzyme system, or they want to inhibit or bind to. They develop. Hit generation means making the compounds and testing those compounds through either a high throughput screening sort of a device or a virtual screening. This is a highly cyclical thing. You make a bunch of compounds. These days, everybody wants to claim it's done under artificial intelligence. I'm old-fashioned. I think the organic chemist and the medicinal chemist are much better at coming up with compounds. Computers just spew out lots of things, some of which are impossible to make, some of which are obvious to make. And the medicinal chemist is a little bit smarter about that. From that work, a lead compound is identified, which becomes ultimately a drug candidate and goes into the ADME, absorption, distribution, metabolism, excretion models, and also in animal models, just to show that the drug does what you think it's going to do. If you're developing a drug to bind to a particular enzyme or to inhibit a particular system in the body, you develop a model to show that it does exactly that. And it can be a model in vitro, which means in a test tube, but more useful are models in vivo, because in a test tube, you would introduce the drug right to the model, right to your enzyme, and yeah, it binds really well. But in the humans, maybe it never makes it to that enzyme. Maybe it gets metabolized first or cleared by the liver through metabolic process, or binds to fat and is never seen by the enzyme, by the model system. At the end of that, when you've completed this work, you file an IND, an investigational new drug application with the FDA, to enable you to do clinical trials. And you do the trials. I'll elaborate those in another couple slides. And then you submit it to the FDA, to the European medical agencies, to all of the other parts of the world you want this product to get approved. You ultimately get approval, and the work doesn't stop then. Then you can continue with your post-approval work, which might be to develop a different indication or a different dosage form for the drug. Focusing a little bit more on what goes on in these pre-IND stage before you've filed an IND, the chemist has to make the drug. They have to make it a couple different times, find a route that can be scaled up. So the process development chemist gets involved. They do some screenings of different salts that might be more stable or more soluble. They look for polymorph formation, because different polymorphs, especially for orally administered drugs, will have different biologic effects, different solubility, different permeation or absorption. And the pharmacology, the non-clinical work that's happening at this early stage, is work on the pharmacology and looking at how your new drug candidate interacts with the transporters in your body, the things that transport the drug into the kidney to be excreted or into the cell to be effective. There's a whole slew of transporters that have been identified, and we do toxicology studies. Then we file an IND. These don't quite line up right, but you file an IND, you start phase 1 clinical studies, and during the time that the phase 1 clinical work is going on, and I'm going to go through that in a lot of detail in just a second, the chemist is improving their synthetic route, developing a better formulation, because you can't get a formulation. A formulation means a tablet or a solution or a transdermal patch or a nasal spray. You can't develop a good formulation until you know what the dose is that's going to go into humans. You don't know what the dose is until you've dosed a few humans and seen what their kinetics is in the human, and seen what dose you think might translate to an efficacious dose. So it's very much an iterative process here. In the later part of the development, the chemists are making large-scale batches, but before you can file a new drug application, which is what you file to get your product approved, you have to have made at least 3 batches, usually more, at close to full scale, put them up on stability, both as the chemical, the drug substance, as well as 3 to 5 batches of the dosage form, the tablet or the spray, and have those up on stability. And they want a year's worth of stability at the time you file an NDA. So the chemist is always kind of a little bit behind the 8 ball. They need to have their work done earlier than anybody else. You can't do clinical trials until you have drug. You can't do phase 2 or phase 3 trials until you have a final formulation. But on the other hand, you can't develop a final formulation until you know what the dose is. So there's always a bit of a push-pull that's going on. This slide outlines what's happening in the non-clinical testing space before clinical studies and after clinical studies start. So there's a lot of exploratory work going on to make sure that the drug has the efficacy you think it's going to have in animals, has safety pharmacology, which means that you've assured that your drug isn't going to impact other important regulatory systems in your body. You give a drug to lower cholesterol, but it binds to the hERG enzymes and interferes with your cardiac function. Well, it's not going to be a very good drug. You know, you do need your heart to function, or your respiratory system to function, or your GI system to function. So we do all of this safety pharmacology work. Gene tox is the Ames test, and the in vitro micronucleus test. We need to make sure that the chemicals we're introducing are not mutagenic. And then toxicology. And toxicology is done in typically 2 species, a rodent and a non-rodent. So that would be a mouse or a rat, and a dog or a pig or a non-human primate. And it's done in 3 steps. There's a 4-week tox study, a 3-month tox study, and then a chronic tox study, which is longer than 6 months. And all of those have to be done, and we do it stepwise, because the duration of exposure you could treat humans in your clinical studies is based on how much tox coverage you have. And your work on the ADME side is ongoing throughout. So there are a bunch of kinds of clinical trials that I thought I'd just jump into quickly. So going back here, in clinical research, you do phase 1 studies. The dose is the first time you've administered it to humans. Usually that means healthy volunteers, people who don't have any other disease, just to see what the safety of the drug is, what side effects it causes, and what the kinetics of the drug is. How long does it take until you reach the maximum concentration? How long does the drug remain in your body? And so the way we do those is what's called a ascending dose trial. So that there's a single ascending dose trial and a multiple ascending dose trial, or the SAD and the MAD. And we start with a very low dose. These numbers are just examples. It's determined by the kinetic of your product, but kinetics of your product and the safety of your product. You dose a very low dose to a small number of volunteers, 6 to 8 people. And if they tolerate it well, you triple the dose. Usually we go up in factors of 3, but not necessarily. You triple the dose and dose 8 more people, and then triple it again and dose 8 more people, and keep going up in the blue single ascending dose until you've achieved either a side effect that suggests you shouldn't go any higher, or you've gotten high enough that you think you've achieved a multiple of where you think the drug will be effective and you can stop. Buried in this, you also start doing multiple ascending doses. So you take that low dose, you don't have to go down to the lowest dose, but a low dose, and you now treat people for a week and see what happens. And then increase the dose and treat them for a week more. Because you don't know the dose at all, this is usually done with a very easy formulation, so you can increase it. So it's often done with what we call a powder in bottle. The pharmacist at the clinical site is given bottles of the drug, just the plain drug substance, and a solution to dissolve it in. And they can just vary the concentration or vary the volume to do this escalating. And the goal here is, as I said, to figure out what the kinetics are and to identify the safety. Then we move into, I'm going to skip this one in order of time. We then move into phase 2 and phase 3. And the goal of phase 2 is to identify the dose, and the goal of phase 3 is to identify the effectiveness and confirm the effectiveness. And these are usually done as randomized, double-blind, placebo-controlled trials, which means that you identify patients, you randomize them, in this case, in this example, to 3 different groups. It could be 2 groups, it could be 5 groups, but patients are just randomly by a computer assigned to either get the placebo, or they might get, in this particular example, 80 milligrams of drug or 160 milligrams of drug. They're treated for a period of time and then stop the study and go into follow-up for safety. This is used for, as I said, the phase 2 and phase 3 trials. So these are now patients, people with the disease under study, under evaluation, and they're followed for using all of the appropriate tests that need to be done for the particular indication that you're doing. So when you hear people talking about studies, usually they're referring to the randomized double-blind placebo-controlled trial. In phase 2, which is the next phase that you do, you're trying to explore the dose and the regimen. Should it be 100 milligrams given once a day? Should it be 50 milligrams given twice a day? Should it be 100 milligrams given 3 times a day? What's the right dose? What's the right regimen? How many times a day it needs to be given? You also are, now this is your first time you've put the drug into a patient with the disease that you're studying, but the size of the trial and the sample sizes aren't usually large enough or long enough to get a full understanding of the efficacy. It's your time to explore different endpoints, looking at quality of life measures, perhaps. How does the patient feel better on the drug? Is their pain reduced? Can they walk longer distance? It's also a time to do a lot of biologic monitoring, imaging, and measuring reduction in symptomatology. And from phase 2, you hopefully identify the dose, the regimen, and the endpoints that you need to put into phase 3. You can often add in phase 2 biomarkers, which would be things like molecular, histologic, or radiographic measurements of change, like measurement of blood pressure would be a biomarker, or heart rate would be a biomarker, or change in your HbA1c if it's a diabetic disease. Those are all biomarkers. They're not measuring the actual disease progression, but they are giving you information that enables you to understand what's happening in the patient. You can also start looking for target engagement. How well does your drug bind to the target? And in some cases, that's pretty straightforward using imaging or measures like PET scans or tumor markers, just size of the tumor. But one of your things that you're really trying to figure out before you start a phase 3 study is, what's the right dose? Is it binding to the target? Do you get more if you double the dose, or are you achieving a threshold or a saturation? Assuming you've passed through that, you move into phase 3 studies, and the phase 3 studies are the big, long studies. This is where you're now spending sometimes as much as $100 million for a study. This is an expensive proposition, drug development. And the goal of this is to confirm the efficacy and the safety. The regulations require, in general, 2 large randomized controlled, that don't have to be placebo, but controlled double-blind studies. And FDA asks for 2 studies because they want a confirmation that the results can be duplicated by different investigators in different patients. They can be two identical studies or they can be two related studies. And here you're now assessing the regimen that you expect in the patient population you expect to take it with a little looser controls. In phase 2, you really target patients pretty tightly with what condition, what they have to have the disease you're studying that's been documented. They have to have reasonable kidney function and liver function and cardiac function, and it's all pretty tightly controlled. In phase 3, it's still controlled more so than in the real world environment, but it's looser so that you can start to get a sense of what's going to happen when the product is out there in the marketplace. I wanted to say a little bit about blinding. Good clinical studies are blinded, and phase 2 and phase 3 studies, we call them double blind. They're really triple blind. The participant doesn't know what they're getting. The investigator doesn't know what they're getting. The sponsor doesn't know what's being given to any individual patient. Now, obviously, somebody in the pharmacy and in the dispensing group at the CRO or in your manufacturing facility does know who's getting what, because that's how we have to make sure that we know who's on which treatment arm. But everybody else is blinded. And the reason for that is to minimize bias that might creep in unintentionally. I'm not suggesting anyone's trying to commit fraud, but just unintentional bias. Doctor comes in, they don't have any say in which patient gets which drug. That they identify a patient, they identify that they are suitable for the trial, the patient agrees to be in the trial, and a computer assigns them to a treatment group. And all the doctor knows is it's study ID 101, patient 101, and they're to get drug code 1002, and that's all they know. So that prevents the doctor from making a subjective decision of, oh, this patient's kind of sick, let's put them in the active group because I want to make sure they get an active treatment versus a placebo. That's eliminated. It also eliminates assessment bias. A lot of drugs are approved based on pretty subjective measures. Either the doctor saying they think the patient's condition is getting better, their symptoms are getting better, or the patient saying their symptoms are getting better. Pain trials and dementia trials and depression trials, all of these indications are very hard to measure. You can put together questionnaires of how quickly someone can do a cognitive test, but a lot of it's about, does the patient feel better? They'll ask you, what's your pain on a scale of 1 to 10? How many times have you been asked that? Well, what my 10 might be versus what your 10 might be is very different. And what my 10 might be today might be different what I say my pain is a scale of 10 tomorrow, because maybe my pain is worse and I didn't realize it could get worse, but it did. So again, having a blinded assessment eliminates some of the bias around that. And then there's something called the white coat bias, which is a bias about patient reporting things that the patients want their doctors to feel good about them, that they're good patients. They're responding well. And so that they might, especially for these patient reported outcomes, might minimize their symptomatology. There's always a mechanism, though, if something happens to a patient in a study and they need to know what the patient is on, they've taken a whole handful of their drugs or they're having a really bad side effect, there's always an ability to immediately unblind the patient. The doctor can do that. The sponsor can do that by logging into a website or calling a phone number and getting the assignment, so that there is information there as safety for the subject. So when you've done all that work, you submit an NDA. And in today's world, these are done electronically. When I got started, they were done in paper. When they were done, and they're huge documents, huge compilation of work. The typical size is measured in the megabyte, and it's huge number of megabytes. You submit, push to submit on an electronic system, and it will take sometimes hours for it to transmit through the system. Back in the old days, it was hundreds of thousands of pages. We did one NDA, and they were submitted in duplicate. So we did one NDA that was when I was in a company. It was a small company. It was the first company's first NDA. We hired a truck to drive the boxes across the country to deliver them to the FDA. And we made the truck drive through a tape where all the employees were out cheering as the drug passed the finish line to go to the FDA, or maybe the starting point. And the reason they're huge is that we submit not only the study reports, but we submit the raw data as well. So this triangle, it's called the CTD, or the Common Technical Document Triangle, and it's divided into 5 parts, really. I'm going to start at the bottom. There's a module on this quality data. That would be the chemistry, manufacturing, control data. That describes all the batches that have made, includes the batch records, and includes the stability data from all of, not just the report that summarizes it, but the raw data itself for all of the stability batches. There's the non-clinical study reports, which includes all of the study reports that have been done on animals and in vitro. This is often several hundred. And for the tox reports, again, it includes the actual data of every single animal that was tested, what their body weight was, how much food they ate, and what their symptomatology was, and then what happened on the histopathology for the organs and the slides to measure it. Then there's the clinical data, and this is the clinical data on a small NDA for a rare drug would be about 500 patients. A large NDA for something like diabetes or heart disease or hypertension would be 30,000 patients, or viruses, antivirals, or vaccines. It's even more than that. And all of those data, again, are submitted in numerous forms, both summarized, so you can see the mean of the patient, but also all the way down to the individual patient and what they reported. The other parts of the NDA, this module 2, is summary documents where you summarize the quality information is summarized, the non-clinical information is summarized. These are about 50 to 100 page documents each, all 5 of them. And those are for the other reviewers. So if you are a reviewer in toxicology, you do need to know a little bit about the drug, so you can review the quality summary to find out what the drug is, what the polymorphs are, what the excipients are, what the stability of the drug is, what the impurity profile might look like. You care about all that stuff. Or if you're the clinical reviewer, you might want to know about the tox, you might want to know about the CMC. All the reviewers have access to all of it, but these module 2 summaries are to pull things together, tell the story of the drug. This triangle, this summary, this system has been harmonized so you can use basically the same body of information for most of the countries in the world, for Europe, for UK, for all the countries in Europe, for most of the South American countries, for China, for Japan, not so much for the African countries yet. They use the WHO approvals usually. And most of it can be submitted in English for all of those different countries in the world. The only part that is regionalized is the forms. Every country has its own form, and the information that you're going to propose to give to the patient, so-called what we call patient-facing information, the package, the description of the product, any advertising you might want to do, any instructions for a patient on how to use the product, those all have to be in the local language, because we don't expect all of the patients to read English, but we do expect most of the reviewers to read English. most of them. Some countries require some of the clinical reports in particular to be translated into the local language, but mostly the review is done in English. The FDA review time for an NDA is 6 to 10 months with all that data. And at the end of that time, you get either an approval or a list of questions. Of course, you get questions all along the way. And the information contained within this whole system is kept confidential until the product is approved. Once it's approved, the FDA publishes a review summary of what they considered, and that's all available online. So what do we expect to see? So the approval criteria has to show that the risks, the benefits outweigh the risk. Everything here is a risk-benefit discussion. There's safety signals in every drug out there, and you need to be able to show that the benefit from a clinical outcome or a validated surrogate endpoint is the benefit is better, is higher, greater than the risk. And the FDA, the labeling has to be appropriate. So the FDA's primary way of communicating their review and their assessment of your drug is through the package insert. And that's going to be the rest of this talk. Am I done? Should I be stopping now? Do I have a little more time?
I think you can have a little bit more time, maybe 5 minutes.
Okay, well, then we're going to have to gallop through this. I'm going to skip this slide, and I'm going to skip the generic slides. And I'm going to talk about the package insert. So package inserts are the way to communicate FDA, and I want to talk about how they're designed, how they're authorized. I will share the slides with anybody who asks. But a package insert, if you look at the screen, you know, it's that little piece of paper that's folded up on top of your thing, sometimes in like tissue paper, and it unfolds to an enormous document. The top of the package insert is the highlight section, which is shown here for one product, but it contains the information they really want you to know. And so here is an example of a particular package insert. This particular example I picked because it has this bit identified in a black box. They don't all have that, but that's called a black box warning. So if there's a drug that has a really nasty side effect or needs particular monitoring they want the doctors to know about, they pull it right up there front and center and put it in bold in a black box and say, watch for that one. The rest of the information is just a summary of what's in the rest of that package insert. And I'm going to skip this one. I'm going to skip this one. FDA is focused primarily on safety. So any advertisement or any promotional activity will have a safety part called the important safety information that scrolls through your screen that you can't get rid of. You can't click to make it go away because they want you to read it. And these language will have been negotiated with the FDA for exactly what needs to be published in that important safety information. There'll be a clinical studies section that describes the clinical studies. And then for products, most products, there's a patient package insert, which describes the indication and safety information in non-medical language. And if it's a device, there'll be pictures and show you how to assemble and use your device. And the last thing, maybe this will be my last slide, okay? I want to talk just a little bit about safety because safety is very important. And safety is a numbers game. If you've only treated 300 patients, which was a smallish NDA, and you have an adverse event that happens at 1%, you will have only had 3 cases of that adverse event. And if it happens at less than, at 0.1%, you might not have had any cases. As the numbers get higher, 1,000 or 10,000, you have a few more cases. But when you've got 1 to 5 cases of an event in a patient, you don't know if the adverse event was due to something in that patient's history, some other drug they're taking, a combination of drug, or the drug you're taking itself. And this is the diagram that European Medical Agency uses. You have the tip of the iceberg when the product is approved. You don't know what's happening underneath the water in that iceberg because you don't know what's going to happen when the drug's out there in the general population, when now tens of thousands or hundreds of thousands of people are taking your drug, and the percentage, of a tenth of a percent adverse event in 100,000 patients, there's going to be a bunch of them. So I generally, in my own life, with me and my family and my parents and my grandchildren, I don't like to be the first person taking a new drug. Now, maybe if I had a really nasty disease, I'd feel differently. But if it's for something treatable, take the old tried and true drug. You know what the side effect profile is for that and what your risks are. Don't be the guinea pig. And I will skip that. This is all right, one more slide. Clinical trials are all listed on databases. So if you have a condition that you want to get in a clinical trial for, you can go to clinicaltrials.gov, searchable by indication, by location, by type of drug. I searched on idiopathic pulmonary fibrosis in California. And if you happen to have idiopathic pulmonary fibrosis, this is just 2 entries. There were 64 of them. I can't get them all on one screen, but you could click on this to see what this drug is, where it's being treated. You could see what this drug was, and what the data were. And it's a way of finding trials for yourself, either to take if you're interested in them or to understand what the research might have been. Great tool for researchers too. Okay, I'm sorry. I always go long, so I stopped.
That's okay. That's all right. It's a fascinating topic. And I actually have one question that I'm interested in this first part that you talked about. You said that doctors can prescribe a drug that's already been approved for anything else, okay? But what I'm interested to know is, okay, they do that, but then do they have to get back with the FDA so the package insert gets bigger?
No, the sponsor of the drug can request to have information added to package inserts, and package inserts change regularly. The sponsor wants to get, and the FDA want to make sure if a new safety signal comes in this, the end of the iceberg part of it, that something starts coming up as a safety signal, they'll update that to the package insert. Or if the sponsor wants to add an indication because they've developed a better indication, they can submit a supplemental NDA to add something. But if a doctor just does something, they can report if an adverse event occurs. In fact, doctors, pharmacists, patients, family members, anyone can report a suspected adverse event. That was one of the slides I skipped. But they aren't obligated to. And if they give the patient something off-label, maybe they'll write a little case report up in the medical literature. Maybe not. Don.
Actually, I wanted to add a comment on her comment first. I worked in the field, and if I ever heard anything, I actually was required to report it.
Yeah.
So if someone just made an offhand comment in the supermarket line about my drug, I had to report it.
Yeah, if you get at a picnic and your neighbor says, "Oh yeah, you work on that. Let me tell you, my sister-in-law took that and stubbed her toe," you are obligated to report it within 24 hours.
Wow.
Yeah.
Hi. I have a question about the phase. When I worked in hemophilia, we did phase 1, 2, and 3, but gradually as we morphed out into other things, we did a lot of what we called phase 1, phase 2. Okay. I hated that. Okay. But what I've noticed with something that I'm not working in, which was like gene therapy, is the number of patients are very small. So let's say 100 people in the world. So if you treated 20 people and then you went with the next phase, you would treat everybody in the world. So I had the impression they were skipping phase 3.
Yeah. I tried to present, so I'm trying to present all of drug development in one 40-minute talk, so I had to do some summarizing. Yes, companies play all kinds of games with the numbering of the phases, and FDA doesn't care. They're just looking at the data that's available. And for the example where you'd be treating everybody in the world, they will work with the company to come up with a reasonable size of the studies, a reasonable outcome measure, and a reasonable safety database that would allow them to make regulatory decisions. The newest trend right now is what they call adaptive designs, where they'll do a phase 2 type design, looking at 2 or 3 different dosage strengths and regimens. And then in the middle of that study, have a review committee that unblinds a small number of people, does an interim analysis and says, oh, okay, out of those 4 groups, keep the placebo, but only the mid dose is working. The high dose is too toxic and the low dose is sub-effective. And then that will morph into a 2-arm study that becomes a registration phase 3 study. So there's a lot of flexibility around what's there that I kind of didn't cover because it's not enough time.
I want to let you know, everybody who wants to participate in the contest, that Michael Chang has decided to give you the questions. So he's already given you, I think, question number 1. And remember, you have to reply to Michael. Do not hit reply all. Reply only to Michael, okay? And
Is this
that
by email or is this on the screen
in the
here?
chat, in the chat.
Oh, no wonder why I can't find it.
Yeah, in the chat, okay? So he's put in question number 1, and this is the way that you will be able to win a prize if you get all the questions correct. And it's all based on what Natalie said today. So you don't have to have any other special knowledge.
Michael, you had your hand up. Was that what you wanted to talk about?
That's correct.
Okay. Anybody else with questions?
Well, while we're waiting, I just want to say that I put a link in the chat to the event we're doing at Lincoln Elementary School next Tuesday evening in Richmond. So if anyone happens to have some time that evening and would like to join us, actually at 5 p.m., we'd love to have a couple extra volunteers there. You can contact me or Michael. So again, the link is in the chat.
So I see question number 1. Are you sending out question 2 and 3, Michael?
2 is just sent.
Okay. Yeah, Michael is the one who comes up with all of these questions. And, it's hard to do while he's listening to the lecture. So, but he has an advantage because was trained as an organic chemist. So a lot of this, a lot of what Natalie did was organic chemistry, and Michael understands that sort of thing.
I wanted to comment on one other aspect that was just mentioned, which is the adverse events. And the FDA is very thorough, apparently, as we know, about collecting information regarding adverse events. And it could literally be, as we were just told, that someone stubbed their toe after taking the drug or whatever it might be. And so the FDA collects an enormous number of these reports, and then they have to go through them and figure out if any of them is really connected with the treatment and if any of them was really caused by the treatment. And so most of the adverse events turn out to be irrelevant, and it takes them time to determine all that. But if there's something relevant, they hope they will find it. But we have to understand that adverse events mostly are not significant. The fact they're reported it's just the starting point for the investigation.
Yeah. And, and almost every drug label you look at, there'll be an adverse event around nausea, an adverse event around vomiting, and an adverse event around respiratory infections, because people get colds and flus and things while they're on the trial. So while the questions are coming in and people are answering them, I'll put one more thing up that covers adverse events. This is the FAERS dashboard. There's also a VAERS dashboard, which is for vaccines. But this is the publicly searchable list of adverse events. I've looked at it in March of 22, so this is a little old, but at that point in this database, they had over 25 million reports, 24 million reports, of which 13.5 were serious and 2 million were death. And you can search it by drug, which is really kind of cool. If you're taking a particular drug, you'll have to log in and play with it. But I did this search in 22 on, which was middle of the COVID era. Remdesivir was the drug, that was being given at that time to treat COVID. And you could see the adverse events that were reported for this drug. Most of them were liver function, elevation of liver enzymes, although there were a certain number of deaths, but who knows whether those deaths were due to the drug or deaths due to the COVID, right? COVID kills people. Some of them were given off-label. There were some bradycardia, slowing of heart rate. You can search it by age group. You can search it by how many people were hospitalized or had another outcome. And the other cool thing that I don't have a slide in for is you can put 3 or 4 drugs in at the same time. So if you wanted to look at statins and you are being prescribed one statin, but you're worried statins are for cholesterol, and the side effect, one of the more troubling side effects of statins is that they can cause muscle weakness and muscle pain. And so you can search 4 or 5 different statins and look at the incidence of muscle pain if that's causing you problems and you want to see how to perhaps suggest to your doctor what drug to give, or the doctor can do this if they want to get the data. So it's kind of a cool thing. It's also been the vaccine one, the VAERS data board has been used a lot by the people who want to tell you that vaccines are dangerous. And they search that and then they see all of the side effects that have been reported from vaccines, and then give you my medical prejudice, my political prejudice is showing, but then they give you the misinformation about vaccines. Okay, I'll stop.
I wanted to also say that I agree with Natalie's recommendation about how she prefers old drugs. I know from personal experience that I have taken some of the oldest of the old, and I I know I felt more comfortable taking them. And you know what? Those old drugs work. So I don't.
They're cheaper too.
Oh,
Yeah, they're
much,
cheaper.
much. I agree.
Shoshona.
The only objection I had to using old drugs, I actually studied a lot of patients with TB. And a lot of the drugs that were developed in the 1950s, people are still taking. And they're very hard for it to take. And, better drug development on this whole field. And, I was just told basically they can't make money off of it. So.
Well, and the TB has evolved as well. I mean,
yes,
you don't want to take old antiviral drugs either. So my recommendation was not across the board.
Okay. Yeah. And the other thing that I worked in critical care, and we were always running some kind of drug study. And it's very interesting, even in a blinded study, it's very actually hard to really do a double-blinded study. We did it on magnesium drips, antithrombolytics, melanoma treatments, you name it. And what was curious about it is that actually we could see where there was sometimes effect. And then the other time, what was hard is that we never knew what to follow up. Sometimes the studies were just abruptly ended. And so we never knew kind of what was happening. But it was pretty especially like with when they were using a lot of stents and then using antithrombolytics because the vessels will actually proliferate when there's damage to them. So it's funny. I've been to conferences because of they don't want the vessel to close again, so there's increased bleeding and the amount of bleeding that we were all dealing with. And basically, nurses used to measure it by, as they're putting pressure down, how much blood was inching up their wrists. And I said, someone needs to actually look at this. This is too much blood. So, but it's very interesting. We rarely found out the outcomes of a lot of the drug tests that we were in.
They're supposed to tell you.
Mm-hmm.
And they're supposed to give the outcome data by patient to the treating physician, to the PI in the study. They don't always do that. They're supposed to, but yeah. And nowadays in clinicaltrials.gov, the sponsor is required to post the overall outcome, not the individual per patient outcome, but the overall outcome. And that's one of the benefits of clinicaltrials.gov is that sponsors are obligated to report both the positive outcomes and the outcomes of the failed studies that maybe don't get published as quickly in journals as the good ones.
Fanny?
Yeah, I just have a quick question when it comes to this, since you've seen the industry shift from before to more technologically advanced now. When it comes to these databases and these announcements and other things like that, I was curious, where would these be posted? Because now it seems like you have the world on your fingertips. You can look up all the medicines that you or your family members are on, but before that, was it always that easily accessible? Or is this just newer just because we have the technology to share all the information now?
Yeah, it's been, the industry or the pharmaceutical industry and the FDA have evolved as with everybody else. FDA's reviews and I gave you in the slides I ran through at the end, a couple websites that where you can get the FDA label and the FDA review. Those have been up since the 90s. So they're the really old drugs are harder to get. You have to get that through Freedom of Information Act, but the modern drugs are there, and there's an NIH website as well. Maybe I'll share again and go back to that.
Well, Natalie, you have shared all of your slides. So if any of the audience members are interested, just contact the California section, WCC, and I can get you the slides for you.
And or contact me as well. I would be happy to give them. These at the bottom of this slide are two good websites to get label information. Drugs at FDA, which is a database that of any drug FDA has approved, and then Daily Med, which is done by the NIH, is also a really great website. And then corporate websites. You just Google Januvia and you'll get all the information in the world you wanted on that. And if you click, I always click on the corporate website that I'm a healthcare professional. I'm not a doctor, but I want to know the information. And that gives you the information they're telling the doctors. Mari.
Yeah, so thank you so much for this great talk. And I have one question that maybe not good question. So the placebo, for example, what are the placebos? So just technically, so if we have a new disease, for example, unknown, the very new disease, we don't have the old drugs, nothing. So what are the placebos? So I don't know that. So
the placebos, they're usually the formulation minus the active. So there's two ways of doing placebo-controlled trials. One is just nothing. You give the patient, you make a tablet that has your active component in lactose and magsteroid and microcrystalline cellulose binder, and the placebo is just a tablet minus the active. That's one approach. And the other approach, if it's a disease that's really serious and you can't put people on no treatment for a long period of time, you blind the study by giving everybody what's called the standard of care. So let's say it's a cancer treatment. You're treating patients for cancer. You give everybody what the medical community agrees is the appropriate treatment for that particular cancer. And then everybody, active and placebo, and then you randomize people to the standard of care plus your new treatment or the standard of care plus a placebo, so that you have two groups that you can look at with and without your treatment, but you're not putting the patient at risk of having no treatment at all for a period of time, which if it's a cancer or, a disease that progresses vigorously and actively, you have to treat them with something.
Yeah.
Okay, thank you so much.
Mhm.
Are there any other questions? If not, then let's just thank Natalie, and I'm so glad that you agreed to volunteer to do this, Natalie. It's wonderful. Thank you.
My pleasure.
Thank
Thank
you.
you.